So, my name is Lana Haganovich, and I'm going to be presenting a study that looks at the influence of the diamondback moth's gut microbiome on its herbivory. Herbivory being defined as the capacity of the moth to consume plant matter and function as an herbivore. Here is the title of the study: Metagenomic sequencing of diamondback moth gut microbiome unveils key holobiont adaptations for herbivory. So to start us off, I'm going to give a little bit of background information. So the diamondback moth, the scientific name being Plutella xylostella, is one of the world's most destructive pests, and it's very resistant to insecticides. It feeds on cruciferous vegetables like cabbage, broccoli, cauliflower, and it's a globally known massive pest in the world. It even co-evolved along with the cruciferous vegetables, which shows how dependent it is on these vegetables, which, of course, people rely on because of our agricultural needs. So these moths spend their entire lives around the host plant. They lay eggs on it, and the problem being that they're resistant to most insecticides. So the authors of this study actually did a previous study that showed that the moth has the ability to detoxify plant defense compounds, which help it work as a pest and keep feeding on those vegetables, because plants, of course, have certain chemical compounds built into them that serve as defenses. But certain insects are able to bypass these defense mechanisms. And this study is specifically looking at the diamondback moth and how it's able to do this. Certain gut bacteria play a really important role in the breakdown of plant cell walls, the detoxification of plant phenolics. Phenolics are certain defensive compounds that plants have, and the synthesis of amino acids, which work to nourish the moth itself. But this study concludes that the previous study that the authors did was actually insufficient, and this study will actually go to create a more complete taxonomic and functional profile of the gut microbiota, meaning it's going to break them down into taxonomical units, classify the microbes into those taxonomic classifications, and look at the functions of those microbes and the enzymes that they have, and the mechanism by which they work to promote the herbivory of the moth. And the study is specifically going to be using metagenomic sequencing. So some enzymes support amino acid synthesis, others support digestion and detoxification functions. But this is really important because if the study finds certain mechanisms that work towards making the moth herbivorous, that can actually be a target for future research to try to figure out how to, target these mechanisms and make them less able to proceed, and therefore stop the herbivorous attack of the moths on crops and help farmers and people who need the agriculture. So past studies have proven that other insects have these capabilities, like termite gut bacteria helps them break down cellulose, which is in plant walls, and obviously breaking it down is bad for the plant. Gut bacteria in honeybees helps them biosynthesize vitamin B12, which helps the honeybees themselves, and degrade pectin. Pectin being a substance in plants, so degrading the pectin serves as eating the plants. And bean bugs actually have a symbiont called Burkholderia, which helps the bean bug degrade an insecticide known as phenytoin. And this study is specifically going to be looking at holobionts, and the holobiont is known as a term that holobiont is a term that defines the unit of the individual animal and all of the microbes in and around that animal. So it treats it as a single unit, and that's mostly what the study is going to be looking at in these moths and the gut microbiota. So because of what we know about the diamondback moth, the fact that it's resistant to so many insecticides, the fact that it's such an awesome pest, this researchers decided that they should look at the gut microbiome and the role that the gut microbiome plays in the herbivorous mechanism. Because, like I said, this could provide like almost a chink in the armor for that microbe, for the moth, which for future research to attack it and then try to suppress these capabilities. The thing is, many previous studies have looked at bacteria in the gut, but they usually used culturing, which is when you multiply microbial organisms by letting them reproduce, like in a medium, or denaturing gradient gel electrophoresis, DGGE, which is a technique that separates a mix of DNA fragments according to their melting point. But the authors of the study determined that the disadvantage of these techniques is they only partially demonstrate the diversity in the gut microbiome, but they want to look at it holistically. They want to get a full, complete taxonomic profile of the gut microbiome in these moths. So, therefore, instead of using the methods of their previous study, which is where they sequenced V6 region of 16S rRNA, they actually ended up using metagenomics. And metagenomics is particularly special because it looks at all of the nucleotide sequences in a given community of microbes. And this obviously provides a more holistic view. So they're looking at the hologenome, which encompasses the entire genome of the animal plus all the symbiotic microbes. The study specifically wants to look at the coevolution of the diamondback moth's gut microbiota along with the moth itself, because there's a theory that they co-evolved essentially in order to help the moth adapt to its environment and also arise as such a great pest and herbivore. But it also wants to look at what specific bacteria, and although it's going to get a holistic profile, it wants to look at the specific bacteria in the moth's microbiome and which of those play the greatest role in those herbivorous mechanisms. And of course, after that, it's going to pave the way for new research to attack these bacteria and their mechanisms. And of course, the specific bacteria are going to come with enzymes and certain proteins that participate in these mechanisms, and these are going to be the main targets for attack. So onto the methodology used in this study. So first, the insects had to actually be reared. So a certain strain of the Plutella zalistella, which from now on is going to be just referred to as the diamondback moth, were collected actually in July 2004 from a vegetable field in Fuzhou, China. The larvae were fed radish seedlings at 25 degrees Celsius in 70 to 80% relative humidity, and a photoperiod that consisted of 16 hours of light and 8 hours of dark. So 2/3 of their day in light, 1/3 in dark. And most importantly, the insects were given no exposure to insecticides. The pupae, after the larvae matured into pupae, they were collected and then they were put into a 500 milliliter plastic bottle. And then once they grew adults, they began being fed a 10% honey solution, which encouraged them to mate and finally lay eggs. And finally, the neonates, which hatched from the eggs, which at this point were the pure generation that was born during the study, they were passed through holes in the bottle and they dropped onto the radish plants. So 200 individuals each for 3 different developmental stages, one being the third instar larva stage, the other being a pupae, and the other being adults. So 200 from each of these 3 groups were randomly picked and sampled, no matter the sex of the insect. They were sterilized, dissected, the gut contents homogenized with a milliliter of sterile DI water, and they were frozen at negative 80 degrees Celsius for DNA extraction. And so the QI-AMP DNA stool mini kit was used to extract the total DNA from all the samples, and then it was run on 1% agarose gels and recovered. So finally, the most important part of the methodology is actually the construction of the DNA library and the actual sequencing, and this obviously correlates to our class, the microbiome. So a paired end library was created and the insert size of each of the DNA sequences was 350 base pairs for each life stage sample. High throughput sequencing was done using Illumina HiSeq 2000 to obtain the metagenome data. So each of the paired end reads had a length of 100 base pairs. Some of them had an N, meaning that any of the 4 bases could have been there because it was inconclusive. Usually it's caused by weak signal during sequencing. It can also happen if the read length is too long for the technique being used. So these were discarded if they had an N, if they had 3 or more N's, meaning that it was unclear what bases they were, or if there was adapter contamination. So the reads that were contaminated with the host genetic material were also removed because we're just trying to isolate the actual bacteria in the sample rather than the host itself. We're looking at the bacterial genome. And then the rest that were deemed effective, they ended up being used. So a method called the SOAP de novo assembler was used to take the effective reads from each sample, and they constructed a long contig. A contig is basically a set of DNA sequences or segments that overlap so that they give a fairly accurate representation of the region of the genome. The metagene program was then used to predict the open reading frames for the contigs that were more than 500 base pairs. An open reading frame is essentially a length of DNA that is going to end up being transcribed into RNA. It basically is the span of the DNA sequence between the stop and the start codon. And in that span, that's where translation starts and stops. Therefore, that's the part of the DNA that's being translated to an actual protein being assembled. So the metagene program was used to find these quote-unquote functional regions in the longer contigs. And these open reading frames were the ones that were longer than 100 base pairs from all of the samples, so not discriminating on any of the developmental stages. They were picked and they were used to make a non-redundant gene catalog. And then they took effective reads. Effective reads are basically sequences of base pairs that correspond to some single DNA fragment from each sample, and they aligned them against the gene catalog using SOAP2 this time. The requirement was that at least 90%, that there was at least a 90% identity between the base pairs before they could be considered aligned, which led to a high degree of match and a high degree of accuracy. These aligned results were then analyzed for their gene relative abundance. And this was done by just counting how many reads mapped to the gene. And then finally, each gene in the catalog was aligned against the NCBI non-redundant protein database. And this was simply to just reference it against that database to classify the taxonomy. It was compared with the KEGG database, which classifies enzymes and their functions, in order to find the function using BLAST-P. When they ran the BLAST-P, they used a condition of the e-value being less than 1 over 100,000. And this is because a low e-value corresponds to a high degree of significance in the match results, meaning that it was not a fluke. The fact that these results are matched means something functionally. And then finally, this information was then used to find the abundance of genes from the same taxon. Taxon could mean any taxonomical classification, so either family, species, or class, or the abundance of genes in the same KEGG ortholog group. KEGG ortholog groups are classified by function. So this made it possible to create a full taxonomic profile of the microbial community. Now, they were trying to test which bacteria led to actual biomass degradation, and these bacteria would obviously be more significant for the herbivorous abilities of the moth. So they dissected gut contents for 15 fourth instar larvae, fourth instar being a phase of the larval development, homogenized it, and then they did a gut content. They took the gut content and then they suspended it in 50 microliters of suspension, and they spotted it onto carboxymethylcellulose, xylem, and pectin degrading enzyme isolation medium. So the important thing to note is that carboxymethylcellulose, which is going to be referred to as CMC from now on, as well as xylem and pectin, they're all constituents of plant bodies. So these are all basically plant biomass: pectin, xylem, CMC. So the goal was essentially to find whether or not these bacteria can degrade this biomass. And if they can, obviously, being that this biomass is from plants and being that the bacteria are from the moths, that must mean that the moth has the capability, because of this bacteria, to degrade this plant matter. And then the bacteria were identified. So the bacteria that were found to have these degrading activities were purified and named. The names are not really relevant, but they were DBMG1, DBMG2, DBMG3, and so on, all the way to DBMG6. So 6 strains of bacteria were found to have these abilities. The important thing to note is that the way that they determined that the bacteria could degrade this biomass was because they dyed the biomass on the plates. Pectin had one on one plate, xylem on the other plate, CMC on another plate, and they were looking for zones that were visibly clear based on the dye that they could see them, and this is where the degrading enzyme activity was taking place. And then 16S rRNA from each of those 6 strains was amplified in order to identify the bacteria, and they did this by using universal primers, and then PCR was carried out. And finally, I'm going to move on to the actual results of the study. So, this first figure here, figure 1, A and B, it shows the composition of the microbiota in the gut of this diamondback moth. So, you can see figure A divides them by phylum, whereas figure B divides them by species, which is a little bit more specific. But we can see that in figure A that the dominant bacterial group in all 3 stages, larva, pupa, and adult, was proteobacteria. It was 99.1% in pupa, 99.7% in adult, and 94.8% in larva. Firmicutes was next, and then cyanobacteria, and etc. The other ones are not as relevant. So essentially, when you break this up, it shows that there's 10 phyla and 148 species of microorganisms that comprise the entire gut microbiome of the moth. And this was, by the way, pretty close to the previous study with the VS sequencing, which showed the 150 taxonomic units identified at the order level. So within these proteobacteria classifications, there are a couple bacteria that stand out. So if you look at figure B that divides them by species, you can tell that in the larva, Enterobacteria cloacae make up 39.2% of the composition of the microbiome, and then 48.2 in the pupa and 35.5 in the adult. And then after the cloacae is the Enterobacteria aspergillus, which makes up 14.3, 12.6, and then 25% respectively in larva, pupa, and adult. Although the proteobacteria were dominated by the cloacae and aspergillus, the phylum was actually represented in the microbiome by 100 different species. So one thing to note from this figure is that the adults and pupa had pretty similar diversities of the gut microbiota, but the diversity was actually lower in larva, because as you can see, the larva had only 94.8% dominance of proteobacteria, whereas the numbers were above 99% for the pupa and adult. This could potentially be because the food that they eat, like larvae only really eat plant tissue, whereas the adults pretty much eat just simple carbs. The other thing is that larvae have to metabolize carbs that are pretty complex. And the previous study, actually, the one that they did with the DGGE analysis and the culturing, those actually show that the larva guts are more abundant and diverse. But either way, proteobacteria are more abundant in pupa and adults, and then there's a lower abundance of firmicutes compared to the ones in larva. And then Anopheles gambia is another species that's similar in larva and pupa, but it actually differs from adults because they change. That's the adult stage is where they change from eating waste and debris to eating blood, essentially. So this will pretty much tell you the proportional composition for the microbiota, and this is pretty interesting. Now, moving on to the second figure, the authors of the study, they wanted to verify those previous results, and so they used another method called 454 pyrosequencing. So basically, they took the V3 to V5 region of 16S rRNA in all the 3 different developmental stages of the gut microbiome, and they did bar-coded pyrosequencing. So figure A shows the phylum level, figure B shows the composition at the family level, and then C shows the neighbor joining method phylogeny of the OTUs, which are operational taxonomic units. So the phylogenetic tree ended up showing that the microbiota of the moth gut were divided into 6 different clusters, and you can see on the wheel, blue corresponds to the gamma proteobacteria, light yellow corresponds to beta proteobacteria, Green corresponds to alpha proteobacteria. Purple to Firmicutes, which are the first group that aren't proteobacteria. Dark yellow, which looks more like orange, correspond to actinobacteria. And then the pink corresponds to Bacteroidetes. So most of the sequence from the moth's gut obviously fell into either Proteobacteria or Firmicutes. Pupa in adults plateaued, which was showed on the curves that show the diminishing of density, whereas the larvae did not plateau, which this could also explain the low abundance of operational taxonomic units. The larva had a lot more complex gut microbiome than the pupa in adults. And like I said, this is likely because of the diet and the lifestyle changes in the larva. One thing to note when comparing this study to the V6 sequencing study that they did prior is that the lactobacillus and the vibrio which are also in the phylum Proteobacteria, they were 10 times more abundant in the previous study than this one. And this could potentially be because of the developmental stages and the fact that there's anatomical differences between the individuals in the different stages, as well as just individuals themselves. However, I will say that this is definitely a point of weakness in the study, because theoretically there shouldn't be this large of a difference in the abundance between two different sequencing methods. It could also be because of the gut environment having a low abundance because of the enrichment, or different seasons or health for the insect. I will say I think it's a point of weakness for the study that they collected all of these. These are all generations of insects that were bred from insects in one specific region of China, and given the fact that this moth is called a global pest, there's no guarantee that the gut microbiota data in this study from the Chinese moths from the one specific region corresponds to the other gut microbiomes in moths all over the world. So that should be further examined probably in future studies. There just needs to be more examination of which sequencing methods would give more objective analysis of the diversity of the microbiome, because right now we're coming up pretty inconclusive, given that these two methods gave pretty different results. Another thing to know is that honeybees, termites, and mosquitoes have midguts that are also dominated by proteobacteria. But the second one is different. It's actinobacteria in honeybees, spirochaetes in termites, and cyanobacteria in mosquitoes. Whereas in the diamondback moth, it's Firmicutes. And this could also be due to the environmental habitats, the diets, etc. And also the fact that they eat different plant matter. So now we're going to go into the third figure, which actually looks at the KEGG ortholog annotation of the diamondback moth's gut bacteria. So the previous two figures showed the composition based on the species and the class and phylum of the microbes, but this time we're actually looking at the function of the microbes. So as you can see here, gene functions that were annotated by KEGG, the KEGG database, they showed metabolic function was the most abundant. It was around 47%, 48% for each of the three stages of the total function of gut microbiota. And metabolic activities tend to be associated with glycans, carbs, amino acids, vitamins, xenobiotics, terpenoids, and these are all substances that link to digestion, nutrition, detoxification, which, if you recall from figure B of the study, are important when consuming plant matter. So this could be showing that the fact that these bacteria are linked to these metabolic functions also helps in the consumption of plant matter, which helps in the moths being herbivores. As you can see in part B of the figure, just by the way, part B of the figure shows that the carb metabolism was the most enriched. It was about 10% for each of the three developmental stages. And amino acid was the second most at about 9.5% for each stage. And you can clearly see that if you look down for the different metabolism functions, which bars are the longest, carbohydrate and amino acid. This shows that these microbiota, they might have been involved in food digestion and nutrition. And One study, a completely outside one, looks at the Asian longhorned beetle, and the metagenome of this beetle shows that it had enriched pathways for metabolism of carbs and amino acids, and this was super important for the woody tissue digestion of the synthesis and nutrient synthesis. Um, by the way, and one note is that enzyme families in Figure B stands for enzymes of peptidases and protein kinases, and these were also detected in the gut metagenome. One thing to note is that these moths only eat crucifers, and which have really good defensive chemical compounds called glucosinolates, which repel most insects. But however, this moth is special because it can overcome the toxicity of these glucosinolates, and the reason why might be attributed to the xenobiotic biodegradation and terpenoid metabolism, which are also seen in Figure B as being some metabolism functions from bacteria that were in the gut microbiome. The gut microbiota likely adapted to this defense and made them even better herbivores. So the fourth figure looks at the gut bacteria-mediated metabolism of biomass. So one thing to note is that digestion is really hard, or can be really hard for herbivores, because the degradation enzymatically of cellulose is pretty hard. Cellulose is the structure in plant cell walls. So this figure shows that the moth's gut microbiome had a total of 2.5 thousand genes from 6 families that were shown to be able to catalyze the breakdown of carbs and glucoconjugates. So Figure A here shows the degradation of CMC, which, if you remember from before, is a substance found in plants. So obviously being degraded is pretty bad for the plants, pretty good for the moth. B shows the xylan, and C shows the pectin being degraded. So the red words by the arrows, they show the degradation enzymes that are identified from the metagenome directly. And if you look at figures D, E, and F, they're actually showing the degradation of these 3, CMC, pectin, and xylan, respectively, with E. coli used as a negative control. And this is all experimentally validated. So CMC, pectin, and xylan degradation was detected by clearance zones in the process that I described earlier around these bacteria, with E. coli as a negative control right in the center of the plates. So if you see in D, E, and F, you see numbered dots 1 through 6. These are actually the 6 strains that we named prior. So these are all, can be concluded that they're degrading CMC, xylan, and pectin, and the fact that these bacteria were all isolated from the xylostella gut, or the diamondback moth gut, tells you something for sure. These clearance zones proved that the bacteria from the gut of the moth have the ability to degrade this plant matter. So our next figure is going to look at gut bacteria-mediated degradation of phenols, and these phenols are referring to plant phenolics, which are, again, defense compounds found in plants. So degradation of these is proof of the bacteria helping the moth in some herbivorous way. So as you can see, A shows the enzymes beside the arrows identified from the metagenome, whereas B shows the degradation efficiency at 12 and 24 hours after treatment, with the histograms which show the different treatments. So the yellow is the phenol media without the presence of bacteria. Purple is the phenol media with the presence of bacteria. And the asterisks there are supposed to show a pretty significant difference between the 2 treatments, which tells you that these bacteria are very detrimental to the phenols and degrade them, which is bad for the plants. And finally, our last figure is going to examine the biosynthetic pathways of histidine and threonine, which are 2 amino acids, by the bacteria in the moth. So one really important thing to note is that insects that feed from phloem being the substance in plants, oftentimes they can't synthesize certain amino acids themselves. So the host symbiont, they might cooperate with the microbes to produce these amino acids. The moth genome lacks the entire pathway for the synthesis of histidine from PRPP, and from the synthesis of threonine from aspartate, which you can see in the figure, you can see that both of the mechanisms start with the starting material I just named. But the gut bacteria has the entire pathway for this, and the fact that moths are able to keep surviving and synthesizing threonine and histidine shows that they're doing this with the aid of the bacteria because they wouldn't be able to do it themselves. Their genome entirely lacks that pathway. And this shows that the moth and the symbiont probably co-evolved together to produce these lacking amino acids, and this keeps nourishing the moth. The genes participating in the synthesis of these amino acids were found mainly in